Good afternoon, everyone. I'm Jie Liu, a PhD student from Washington University in St. Louis. It's my great pleasure to present our research on genetic determinants of clonal hematopoiesis at this meeting. I have no disclosure. We know that cancer is caused by somatic, stepwise acquisition of somatic cancer driver mutations, and sometimes we can detect the first mutational step in healthy individuals. For example, the hematopoietic stem cells can acquire mutation that allow them to acquire fitness advantage and expanded to detectable clone. This process is called clonal hematopoiesis, or CH for short, and CH can be detected using next generation sequencing in healthy individuals at a low allele frequency. And age is the most important predictors of CH. The frequency of CH carriers increases exponentially with each decade of life. And the most frequently mutated CH genes like DMT3A, TAT2, AXL1, as showing on the left, overlap the most common myeloid neoplasm driver genes showing on the right. And CH is also associated with the risk of hematologic cancer. Taken together, we think that CH is a potential origin of myeloid neoplasms. As we all know, myeloid neoplasms are fatal with 5 years survival rate less than 30%, and the improving the prevention is an urgent clinical need. We already know myeloid neoplasm have a hereditary basis, but our understanding is limited due to the rarity of the disease and the difficulty in distinguishing germline from somatic mutations in tumor samples. We think CH is the potential origin of myeloid neoplasm, so studying the association between germline mutation and CH might provide insight into our understanding of germline predisposition to myeloid neoplasms. Previous genetic association studies have focused on common germline variants and have identified about 30 common genetic loci that predispose to CH. However, the identification of novel germline mutations, especially rare germline mutations, rare variants that generally confer higher risks, is underpowered because of their rarity. So the goal of this study is to identify rare germline variants that predispose to CH and the myeloid neoplasms. We performed a cohort study of the 500,000 individuals with available whole exome sequencing data and SNP data in the UK Biobank. The frequency of CH and the incidence of myeloid neoplasms among people with germline mutation were compared to those without germline mutation. We hypothesized that CH and myeloid neoplasm will be enriched in people carrying rare pathogenic germline mutation in cancer predisposition genes. And for genes that associate with CH, we will validate that finding using external cohorts, including MSK impact and TCGA. We detected SNV and indels using UK Biobank whole exome data, and the average depth is 50x. The germline variants were called using deep variants, followed by joint genotyping using GNexus. We filtered out low quality, common, and non-pathogenic variants. We restrict our analysis to mutation in about 200 known cancer susceptibility genes, then annotate their pathogenicity using ECMG guideline. The somatic mutation were called using METAC2 verdict. We filtered out artifacts, low quality, and germline variants according to previous standard practices. The CH annotation were performed based on curated databases and literature. For large copy number variation, we call germline events using PenSynV and somatic events using a well-established haplotype of your pipeline, MOCA. In the following slides, I'm going to show some preliminary analysis from the 500,000 individuals in the UK Biobank. First, we identified about 8,700 pathogenic germline mutations, and about 14% of people in the UK Biobank carry a pathogenic germline mutation in cancer susceptibility genes. And about 7% of people carry pathogenic mutation in autosomal dominant genes. And overall, about 18% of individuals in the UK Biobank have CH. The distribution of SNV and indels and copy number variation are largely consistent with previous CH studies. We also found that the percentages of CH carriers are slightly higher among people with germline mutation compared to those without germline mutation. And here we tested the association between germline mutated gene and CH by type. The x-axis shows the odds ratio of CH development, and the y-axis shows the germline mutated genes by functional pathway. We found that germline mutation in multiple functional pathways are associated with CH mutation, especially the TP53 telomere maintenance and the RAS pathway. And more interestingly, we found that some gene like CHEK2 and ATM are associated with CH driven by both SNV, indel, and the copy number variation. But for some gene like TP53, only specifically associated with CH driven by SNV, indel, and for some gene like SOS1 are only associated with CH driven by copy number variation. Then we want to look at whether specific germline mutation predisposed to specific CH mutation. First, we looked at the co-occurrence of germline mutation with CH in the same gene. The x-axis shows the odds ratio of a germline carrier developed CH in the same gene, and the y-axis shows the adjusted p-value, and everything above this dashed line is significant. So here we can see some examples showing acquired mutation, mutation like co-occurring in the same gene as germline mutation have a strong fitness advantage. For example, the TP53 germline carriers have higher frequency of point mutation in TP53 and also chromosome 17 loss. And here we tested the association between germline mutated gene and CH mutated gene driven by SNV and indels. We color-coded this plot by odds ratio. So the red indicates positive associations, blue indicates negative associations, and significant findings are annotated using asterisks. And this red rectangle highlighted the association between germline mutation and CH mutation co-occurring in the same gene. So we can see a clear heterogeneity in both the strength of the association and the associated CH spectrum for each germline mutation mutated gene. And similar for CH driven by copy number variation, we also observed like germline mutation are associated to specific CH patterns. And during up to 14 years of follow-up in the UK Biobank, we observed about 4,900 individuals develop hematologic cancer. And here we tested the association between germline mutated gene and the hematologic cancer risk. The x-axis shows the hazard ratio, and the y-axis shows the germline mutated gene, the significant ones. And for these 8 significant genes, 6 out of them, including TPP3, CHEK2, ATM, POT1, RONX1, and BTK, are also associated with SAGE. So then we want to know whether the acquisition of SAGE can further increase the risk of hematologic cancer compared to having germline mutation alone. As we expected, the acquisition of SAGE does increase the risk of hematologic cancer. For example, among TPP3 germline mutation carriers, having SAGE driven by SNV and indels increase the risk of hematologic cancer, and having SAGE driven by copy number variation increase the risk even further. However, for some gene like POT2, having SAGE driven by SNV and indel doesn't increase the risk, but having copy number variation it does. So in conclusion, we observed a high frequency of pathogenic germline mutation and SAGE in the UK Biobank. We found that rare pathogenic germline mutation in cancer susceptibility genes are associated with the increased risk of SAGE and hematologic malignancies. We also looked into specific SAGE mutations and found that germline mutated genes are also associated with specific SAGE patterns. Especially, we saw SAGE that overlaps germline mutation confers strong selective advantage for certain genes. And we also observed for, among germline mutation carriers, the presence of SAGE can further increase the risk of hematologic cancer. So at the end of my presentation, I want to thank my mentor, Dr. Kelly Bolton, our collaborators, and all the UK Biobank participants for their contribution to this study. Thank you for your attention.